All right, so we're going to go through the muscle tissue lecture. I'll just try to zip through it, just so you hear it if you want to hear it. I mean, there's no reason why you should need to, but in case you want to, we'll just go through and fill it up so we can say that it was done. So there are three types of main muscle that we covered over in lecture. And the first of them is our skeletal. And this is a striated type. And this means it's got stripes. And the stripes are because of the overlap of actin and myosin and their arrangement. So when you look through a light microscope, part of it appears dark and part of it appears light, and they're banded patterns. Then we also have cardiac muscle. This, like skeletal, is striated. So the functional parts of cardiac and skeletal muscle are both actin and myosin. And then the third type we have smooth. Okay, and this is non-striated. So there is actin and myosin in smooth muscle, but it is not arranged in a pattern that allows it to have an alternating pattern of stating. Okay, all muscle has the typical characteristics. The first of them is excitability. And this means that the plasma membrane can actually carry a charge, so it can change its electrical state. It can go from a negative potential on the inside to a positive potential. They are all elastic, so they can change their shape and return to their original shape. They are extendable. And they can be stretched. And they all have contractility. So they all can be contracted. So those are the common traits between all muscles. So the function of skeletal muscle basically is movement. Okay. So it pulls on bones, and it allows bones to actually change their position in relationship to the joint. Another thing that skeletal muscle does is the passage of substances internally, homeostasis, in the form of heat generation. Right? So shivering would be an example of this. Now, the structure of a skeletal muscle is kind of like that of a nerve itself. And individual muscle fibers are surrounded by connective tissue. A bunch of those muscle fibers are gathered together in what's called a fascicle, surrounded by connective tissue. And a bunch of fascicles are surrounded together. To make up the muscle fiber. And these, not muscle fiber, the muscle. And these connective tissue layers are called mesia. And so we have an epimysium, which is around the entire muscle. Then you have the perimysium, which is around a fascicle. And then you have an endomysium, which is around the individual muscle fiber itself. Okay. So the endomysium is areolar connective tissue. Perimysium and epimysium are dense irregular connective tissue. So that's how they exist. The fibers of muscle themselves are the cells. That's what the fibers are. They're the cells of muscle. They are the muscle cells. Okay. So when we say that the skeletal muscle fibers, we're talking about the functional part of a muscle, because this is the part of the muscle that actually does the work. The muscle itself is just a conglomeration of a bunch of cells. Right. So all muscle cells or fibers, depending on how you want to call it, they both mean the same thing. They are multinucleated, which means they have several nuclei. I think they have 10 to hundreds of nuclei, depending on the length of the muscle. And what they come from, where they originate from, is called embryonic myoblasts. Okay, the diameters of a muscle fiber can be up to, so the diameters, and you know the diameter is the distance across a circle, is up to 100 micrometers. And the lengths of these individual cells can be up to 30 centimeters. 30 centimeters. That's pretty long. So the parts of a cell, parts of a muscle cell, a muscle fiber, are pretty much synonymous with those of the cells that you studied in basic biology. Okay, we have what's called the sarcolemma, which is basically just the plasma membrane. We have the sarcoplasm, which is just the cytoplasm itself. We have the sarcoplasmic reticulum, which is nothing more than the endoplasmic reticulum. And typically in muscle cells, it's smooth. So it's a smooth sarcoplasmic reticulum. We have myofibrils. So there's our muscle fibers. We have actin. We have myosin. And actin is usually the thin filament. No, it's not usually, it is the thin filament. Myosin is called the thick filament. And they are arranged in what's called a sarcomere. Okay, so that's how they're arranged in the form of a sarcomere. Now the sarcomere itself, it's the functional unit of the muscle cell. So the muscle cell is the functional unit of the muscle. The sarcomere is the functional part of the cell. And we've already said it is composed of actin and myosin. And wrapped around the actin, there are these things called regulatory proteins. Okay, and the two regulatory proteins are troponin and tropomyosin. Right. Now, if we look at this picture right here, this one right here, this is the actin. This is actin right here. It's actually the conglomeration of like three different proteins. The actin itself, actin, and then troponin and tropomyosin. And tropomyosin, what it does is it kind of like attaches to the actin, and it covers up part of the actin. And troponin attaches to the tropomyosin itself, and it's responsible for moving, the troponin attaches to the tropomyosin, and it's responsible for moving the tropomyosin out of the way so actin can do its job. So how they're arranged is they're arranged, both the actin and the myosin, they're arranged in what's called a sequential order. Okay, so basically this means that we have actin, then myosin, then actin, then myosin, then actin, then myosin, then actin, then myosin. Okay. Right. And eventually what's going to happen is at the end of the muscle cell, you're going to have like the actin and myosin, they're going to attach to the plasma membrane of the cell on one side. And then on the other side, actin and myosin is going to bind to the plasma membrane on the other. So when they contract, basically the actin and myosin are going to pull the two ends of the cells together. So the sarcomere itself being the functional unit, we need to classify what actually is a sarcomere. So the sarcomere is an arrangement of actin and myosin. And the structure of the sarcomere, so if we look here, I'm going to circle the sarcomere. If we look at it, we have this thing called a Z line, and we have a Z line. And what those are is the borders. So they're the Z line. Some people call it the Z disc because it is a three-dimensional structure. But that's basically the end. So when you go from a Z line to a Z line, you are talking about a sarcomere. And this is where the actin anchors. So attached to the Z line, we have the actin itself. Now at the center of this Z line, we have this thing called the M line. And I always think M for middle, it's the middle line. It's an M line. And at this M line, the myosin attaches. Myosin attaches to the M line. So the sarcomere, on both sides, we have Z discs or Z lines. And at the middle, we have an M line. Now, actin attaches to the Z line, and it actually projects toward the middle. And myosin attaches to the M line and projects toward the Z disc. Okay? So myosin, but M for myosin goes that direction. A for actin goes this direction. So what this is there's actually a little bit of overlap. Okay, so the myosin and actin are fixed lengths. They're not going to change their lengths. But what's going to happen for a muscle to contract is what the myosin is going to do is it's actually going to pull the actin, over itself, and that's what contraction basically is. But right now we just need to know the parts, these 4 parts of the sarcomere itself. So the neuromuscular junction. So how does a muscle contract? So firstly, it's an interplay between muscle and a nerve. And the thing you must remember is that in order to get a muscle to contract, it has to be told to contract. Now, the only way you can tell a muscle to contract is if you give it a little bit of a shock or you give it an excitation signal. And that's the only way to functionally stimulate the muscle. Okay. To stimulate the muscle to actually contract. Now, where this happens, where the nerve stimulates the muscle to contract, it is called the neuromuscular junction. And the neuromuscular junction is nothing more than a synapse, really. It's the point at which a neuron communicates with a muscle fiber. And that is the heart of the NMJ, the neuromuscular junction. Okay? It is the synapse between the neuron and the fiber. Okay? So this means the axon of the neuron is presynaptic. And the sarcolemma of the muscle fiber is postsynaptic. Now, this sarcolemma, excuse me, the sarcolemma of the muscle fiber is sometimes called the motor end plate, So if you see that, that's what they're meaning. They mean the muscle fiber after the synapse. Okay. Every single muscle fiber in your body, skeletal muscle fiber, is innervated by a neuron. Okay, and innervated means that there's a nerve going to it. That's basically innervated. There's a nerve that goes to these muscles. And every muscle has to have a nerve going to it. Every skeletal muscle has to have a nerve going to it at a neuromuscular junction. It's just the way muscles work. And if they don't have them, they actually start to waste away. So now how does this work? So now we know that there's a nerve that actually has to go to a muscle fiber in order to stimulate the muscle fiber to contract. Okay. So now what we have is we have this complex interaction between a nerve exciting a muscle and the muscle contracting. And what that is called the excitation contraction coupling. Okay. So we've already stated that we have to excite a muscle fiber. Now, the only way we're going to excite this muscle fiber is we have to depolarize it. And so another way we say depolarize is we say it excites it. Excites it, or you can think of it as depolarize. We will depolarize the muscle fiber. Okay. We need to do this because we must fire an action potential. Okay. You thought you were done with action potentials when we got done with nerves, but they always come back to you. All right. So we have to stimulate the muscle to fire an action potential. Okay. Now this action potential has to travel the entire length of the sarcolemma. Okay. So up to 30 centimeters. Okay. And as well as traveling the entire length of the muscle, it actually has to travel into the muscle as well in order to get to the sarcoplasmic reticulum. Okay. The reason we have to do this is because there's an ion that is stored in the sarcoplasmic reticulum called calcium. And calcium is necessity. It is necessary to cause contraction. Okay. So what the action potential does is it signals the sarcoplasmic reticulum to release calcium. And what this is going to do, this calcium is going to interact with actin and myosin in such a way to allow the myosin to do its job and pull the actin. And when the actin is pulled, the muscle contracts. Okay. So combining the electrical stimulation with the release of calcium resulting in a contraction, that is the excitation contraction coupling, just general overview. What we're going to do is we're going to take a look at how that actually works. Okay. So we've already established that signaling begins when a motor neuron, what it does is it stimulates the neuromuscular junction. That's the first step. The first step happens at the synapse. And the nice thing about this is it's always excitatory. It's always going to be excitatory, so it's no big deal. The axon releases acetylcholine. Acetylcholine is the neurotransmitter. And what this neurotransmitter is going to do, remember, when we go back, if you go back to look at the synapse, when we talk about the neurons, the neurotransmitter is going to float across the synapse. And what it's going to do is going to bind to a receptor on the postsynaptic part of the cell. Now, seeing as we're dealing with the neuromuscular junction, it's going to bind to the motor end plate. That's what it's going to do. Motor end plate. Now, when we were dealing with nerves, we said that it could be inhibitory or it could be a stimulatory signal. In a muscle, it is always a stimulatory signal. So you don't have to think about it at all. If it's a muscle and a nerve stimulates a muscle, the muscle is going to contract. So that means the muscle is going to depolarize. So that's it. Done and done. Now, the hard thing is what has to happen in order for the muscle to depolarize. Well, remember, the ion that is responsible for the positive charge inside of a cell, it is sodium. So what it's going to do is it's going to open sodium channels. Okay? When you open the sodium channel, sodium is going to come into the cell, and eventually, the motor input. Sorry, it's going to open up sodium channels and eventually voltage gated channels. Okay, and this is going to cause the muscle to depolarize. Or in other words, it's going to stimulate the muscle to create an action potential. Right? Now, how do I get this to stop? Pretty simple. I just tell the nerve to stop sending the signal. That's it. Okay? So when you stimulate the muscle fiber, as soon as you stimulate it and it generates this action potential, it pretty much stops generating the action potential as soon as it fires one. Okay? Because what you don't want to do is you don't want to have a constantly contracting muscle, all the time. That's not what you want. Okay? And the way we do this, happens is two ways. One, the nerve just stops releasing the neurotransmitter. And the other way is I degrade the acetylcholine in the synapse. Okay, and that's the second way. And what does this is an enzyme inside of the synapse. It's just waiting for acetylcholine, and that enzyme is called acetylcholinesterase. And what acetylcholinesterase does is it breaks down acetylcholine and it makes it inactive. So that's how we terminate the action potential itself. So moving on. So before we move on, this is how we start the excitation part. Now what we need to do is we need to say, how do we propagate it? Okay. So the propagation is the excitation part of this coupling, of the excitation contraction coupling itself. In order to reach the place where the calcium is stored. Remember, calcium is needed for contraction. So basically, all the nerve is trying to do is just trying to get the muscle to release calcium. That's what the nerve is trying to do. Okay? In order to do that, in order to reach the sarcoplasmic reticulum, the action potential has got to travel over the sarcolemma, and then it's got to travel through these things called transverse tubules. Okay. Now, the reason we have to travel through transverse tubules, because if you look here, if you look at this sarcolemma, and you look at the sarcoplasmic reticulum right here, they run in parallel. They pretty much run in parallel. See, here's the sarcoplasmic reticulum is running in this direction, and then the sarcolemma is running in this direction, and they're separated by space. But the action potential can't jump. It can't jump from the sarcolemma to the sarcoplasmic reticulum. So instead, what it has to do is there's special channels. These special channels called T-tubules. What they do is they're basically parts of the sarcolemma that dip down and run right along the sarcolemma. Okay. And what these T tubules do is basically their only job is to carry the action potential to the sarcoplasmic reticulum. That's it. And once they carry this action potential to the sarcoplasmic reticulum, what they're going to do is they're going to depolarize the sarcoplasmic reticulum, just like it depolarizes a normal membrane. Because the sarcoplasmic reticulum, if you remember from Bio 101, the endoplasmic reticulum is just basically plasma membrane. That's all it is. So it's got the same properties of plasma membrane. So if I have this action potential traveling down the T tubule, it's depolarizing the sarcoplasmic reticulum as well. And what that's going to do is it's going to open up calcium channels. Okay. Now inside of the sarcoplasmic reticulum, we are stuffed full of calcium. That means the concentration of calcium inside of the sarcoplasmic reticulum is much higher than it is in the cell. So when I open these calcium channels, the calcium is just going to flood out of the cell. It's going to flood out of the sarcoplasmic reticulum into the cell. Okay. And that's what's going to do. It's going to allow calcium to flow into the sarcoplasm. And what the arrival of calcium is going to do is it's going to initiate muscle contraction. That's all it's going to do right there. Okay. So the active contraction. So what we're going to do is we're going to pause right here. We're going to pause right here because we're already at over 20 minutes. And we will pick it up with the actual active contraction.